All right, guys. Give me a shout if you can see my presentation screen.
Yes.
Yep, thank you. So, welcome back to the plant lab, right? Well, welcome back to 122 after your practical. And speaking of which, you should be able to see your grade. And now, probably by end of today, you should also be able to actually see your correct answers, right? Like and get the like sort of go through the entire feedback. Um, so that has been done, and your lab notebooks has been graded, and everything else actually have been graded up to date. The thing that I wanted to emphasize to you guys is actually about your continuation of your poster presentations, right? So towards the end of today's lab, we will have more time to talk. Um, and as of now, before we begin, are there any questions regarding the feedback you received for your poster projects, or any other question related to how to progress from here onwards? I'll give you a minute or a few seconds, not a minute. Okay, so I do not hear any questions, so I'm going to continue on with today's PowerPoint. But again, I will open up or like provide some general guidelines towards the end of the lab, right? Okay, moving on. So we are going to start talking about plants from today onwards. We are done with the lab component, and alrighty. So we are done with the animal component, right? And we are going to move on with the plants from this point onwards. And the first group of plants we are going to talk about are the non-vascular plants, right? So let's look at this phylogeny for a quick hot second over here. So we are looking at over here the evolution of the entire the tree, the phylogeny of plants. So the green algae, right, are kind of the common ancestor to all plants, right? So so all the plants that we have today, right, are actually the ancestors to green algae are the ancestors to all the modern plants today, right? Modern and extinct plants, and that's what actually I wanted to mention first over here, right? So and in fact certain green algae, are able to tolerate certain level of exposure to the ambient environments, right? And can tolerate certain level of drying, but they're not fully equipped to withstand prolonged terrestrial exposure, right? So and then the earliest group of land plants, right, are actually these three groups, collectively considered non-vascular plants, right? Hepatophyta, liverworts, bryophyta, true mosses, that's actually fairly common species, and a fairly common taxa, and then hornworts, Anthocerophyta. So now I must mention, if when you look at all of these three lineages, they are all considered non-vascular plants or avascular plants. Now, do you guys think that these three lineages are sister taxa? They are not, right? They are not sister taxa. So that's actually really important to understand. You can see those three do not share a recent common ancestor, right? So their evolution happened really independently, right? And then if you look at certain characteristic features, right? So the origin of all land plants can be traced back to about 475 million years ago. And evolution of the cuticle, which is actually a waxy covering around the above ground components, right, is actually a critical feature which help to minimize water loss. Secondarily, when you are on land, you need some mechanism. Like if you have a cuticle, the problem is the cuticle will actually protect the entire above ground plant component, the photosynthetic components, non-photosynthetic, above ground biomass will be protected by the cuticle. But the problem is cuticle does prevent gaseous exchange, right? You cannot absorb carbon dioxide, you cannot get rid of oxygen, you cannot absorb carbon dioxide for photosynthesis, you cannot absorb oxygen for respiration. So that's a problem. So you need to have some sort of a mechanism in the above ground plant, right, to absorb air, right, to exchange gases. The same thing for the root components as well. The gaseous exchange is not just a problem of roots, but if the above ground component of the plant can exchange gases, the roots can actually piggyback on that gas exchange from the above ground parts. So you need to have some sort of a pore, right, within the cuticle embedded around the plant body that actually sort of circumvents the problem of the cuticle. So understand what I'm saying here. Cuticle is important to prevent dehydration, right? That's what happens when you're on the land. The pores are important to facilitate gaseous exchange without undergoing further dehydration. Okay, now, and then,
Sorry, Professor. You
oh,
you were going in and out, so I didn't hear what you said.
oh, let me repeat the last part one more time. Thank you. Give me a shout, just to make sure that I'm not going in and out for others. Are we good? Can you hear me, or is it? I don't know whether it's a Monica's Wi-Fi issue. Others, give me a shout.
I can hear you clearly.
Very good. Monica, don't worry because the recording you can catch up. Sorry, go ahead.
Oh, I was just saying it was clear on my end too.
Yeah, yeah. I will repeat that part, Monica. But if you miss, please do ask back, okay? And so what I was mentioning is that the evolution of cuticle, right, is important to prevent dehydration. Right now, when you have a cuticle, the problem is that will also prevent gaseous exchange. So therefore, the plants have evolved pores, right, on the above ground component to facilitate gaseous exchange. And then subsequently, the problem with these original pores evolved in the land plants is that you cannot control them. They are always open, right? The problem is, what if you want to prevent further dehydration, right? Then you actually wanted some mechanism to close and open stomata, right, as need be. You have to open and close those pores as needed. If the plant is actually dehydrating, if it is water stressed, like in a drought condition, you need to close the pores. If there are no water stress, well, the plant needs to open the pores. So evolution of stomata happened subsequently, and some of the non-vascular plants, two lineages of the non-vascular plants, actually have stomata, but one lineage, the liverworts, do not, right? Okay. Last thing I want to mention is actually the closest green plant, closest algal relative to all the land plants. It's actually the genus Chara, sorry, by the way, it's genus Chara. English name is stonewort. This particular plant is actually the closest living relative of closest algal relative of all land plants. Now, let's take a look at this guy, Chara, or a stonewort, right? It looks like one of those aquarium plants that you might see, right? And then, if you look closely, there are like this kind of gravel-like things all over the plant body. If you zoom into them, right, it looks like this. If you zoom further in, it looks like that. Those components that are actually sort of granular are actually the reproductive units, right, of that particular algae. Now, evolution of these very unique reproductive structures, right, are actually an evolutionary advancement that you observe in this green algae. And if you look at this picture, you can clearly see this is not like a floating green algae. It is not free floating. It is not just floating on the surface. It's actually anchored. They have a hold fast that will help the algal plant body to anchor to a substrate, right? Now, let's talk about some basic features of the plants and then try to understand whether they are unique or whether that particular characteristic is shared with other taxa within the living domains. Now, plants are So let me go back. Plants are eukaryotic, right? They have organelle, membrane-bound organelle, and they have a membrane-bound nucleus. Autotrophs, right? They can actually photosynthesize, right? Well, not all plants can photosynthesize. There are parasitic plants that cannot photosynthesize. The parasitic plants that have either inactive chloroplast genes, right, or have no photosynthetic pigments in them. So, but most of the plants can photosynthesize. So they are autotrophs. Cell wall, right? Plants have a cell wall. They are multicellular, right? There are no unicellular plants. There are unicellular algae, but there are no unicellular plants. All plants are multicellular. They have mitochondria, have chloroplast, connects back to the autotrophy. So eukaryotic nature is not unique for plants, animals, fungi, and some protists are eukaryotes. Actually, all protists are eukaryotes. Autotrophy, well, largely that's a characteristic of the plant kingdom, but there are certain protists that are able to perform photosynthesis, right? And some archaea and some bacteria are also photosynthetic. So it's not 100% unique, but a very remarkable characteristics of plants are to be the ability to become photosynthetic. Cell wall, there are several other groups like fungi, for example, that have cell walls, but the composition of the cell wall could be different. Multicellular nature, you all know that there are so many other like animals, fungi, all animals, right? Some fungi, actually most fungi and protists, some protists are multicellular in nature. Now remember, if you were wondering about a fungi that is not multicellular, some parasitic fungi are actually unicellular. Yeast is unicellular. In fact, certain fungi can have both unicellular and multicellular forms. That's actually some added information for you. Mitochondria, well, all eukaryotes, right, have mitochondria. Chloroplast, there are some protists that have chloroplast, but bacteria and archaea, even those that are photosynthetic, do not have chloroplasts, right? Now, there are arguments about how some protists actually have chloroplast. Is it by ingesting plant material and then retaining the chloroplast but digesting the rest of the plant material? Is it some sort of a evolutionary symbiotic relationship? For certain protists, it could be, but there are many protists that we don't know how exactly they acquired chloroplasts. But for the moment's sake, be aware that protists could carry chloroplasts, so they could be photosynthetic. And just to make an interesting fact, because of the ability for certain protists to have chloroplast or produce chloroplasts, they could actually have photosynthetic ability as well as heterotrophic ability. So essentially, they are dual function, right? If sunlight is available, they will photosynthesize. But if there are other food particles available, they will also eat that food, right? So because of that, most protists that are photosynthetic are also heterotrophic, right? They can be predators. Moving on, what kind of environmental problems plants encounter? Well, water loss, because they are on the land, right? They are not in water, unlike some of the unlike the algae. So they could face dehydration, water loss. Anchoring. Well, they need to actually they should have a mechanism to root themselves somewhere, right? If they're actually on the land, they cannot free float. If you're in water, yes, some aquatic plants can actually afford to be unanchored and free floating. Most algae are actually free floating, right? They are not anchored. But now, as a land plant, you should have a mechanism to root yourself, anchor yourself. Gaseous exchange. Well, you got rid of the need to so you got rid of water and then you are now on the land, right? Well, then you need to have a particular mechanism to exchange gases, absorb carbon dioxide, absorb oxygen. Obtaining water and nutrient. Should have a mechanism to absorb water and nutrient, because now as algae, you can absorb things through cell wall directly, right? And you are in water, so you can actually directly absorb water and nutrient as you need, right? But if you are in the air exposed to the atmosphere, well, atmosphere does not have an overabundance of water. Soil does not have an overabundance of water or minerals. Have to have a mechanism to absorb those. How do you maximize interception to solar radiation for photosynthesis? That's a challenge. But if you actually have, if availability of water and mineral and food synthesis is actually a challenge, then once a plant synthesizes food and absorb water and minerals, the plant should have a particular mechanism to redistribute that. So they need some sort of a network, is some sort of a redistribution system, a transportation system to distribute matter and energy. And last but not least, storage, right? If you cannot always absorb stuff as needed, right, you need to have a mechanism to store the food you produce, to store the water that you absorb, to retain the minerals you absorb, right? So need for storage, becomes an important concern. So the problem with, why is it a problem? Well, because why water loss is a problem? Because now you are exposing yourself to the environment, you can dry out. So the solution is the evolution of a cuticle, a waxy layer. Quite literally, it's wax, right? That is covering the exposed parts of the plant body, right? In fact, certain, not only the above ground biomass, in certain plants, even the roots tend to have sort of a waxy external covering. Now, secondarily, obtaining water. Well, since algae were living in water, right? They are surrounded by water. Obtaining water was not a problem, right? Now you are on land as a land plant. You cannot directly absorb water, even when it is raining, right? Or even when it is when it has flooded, you cannot absorb water because the cuticle, the waxy layer, not only it prevents dehydration, it prevents absorption, right? So you need to have a particular mechanism like root or root-like structure to absorb water, to absorb minerals. Gas exchange, waxy cuticle will not only prevent water movements, it will also prevent gas movements because wax is impermeable to gases. So you need to have some sort of an opening in the plant body to exchange gases. So hence the pore evolution became a solution. Nutrients. Well, if you are not in water, you are not going to have direct access to nutrients. So you need to have another mechanism to absorb it. Again, roots help water absorption and nutrient absorption as well. Anchoring. Well, again, root-like structures, rhizoids in particular, or actual roots, can actually help plants to anchor to a particular substrate. So, just wanted to underscore these three important steps in plant evolution. First, origin of land plants, right? And those early land plants shared a common ancestor with green algae, right? And now remember, not all algae are green. There are red algae, there's brown algae, right? But green algae were the closest relative to land plants, and the land plants shared the recent ancestor with green algae. Now, number two, origin of vascular plants happened, about 425 million years ago, right? It's way after, so several other non-vascular plant lineages evolved before that, right? Now, what is vascular? Vascular means actually having a specific pipeline system to deliver water and mineral as well as to deliver food, right? Now, we will talk about those vascular systems later on. Number three is actually the latest innovation in land plants is actually that happened about 300 million years ago, is evolution of the seed as a mechanism of reproduction. Right now, understand that all of these are land plants. All of these are land plants, right? And the land plants have two major components: vascular plants that we are not going to talk today, and non-vascular plants. And then vascular plants has two components: those that can produce seeds, or those that cannot produce seeds for the purpose of reproduction. Now, another unique feature that I want you to understand that is important for all the form of land plants is the alteration of generations. What do you mean by alteration of generations? So all land plants alternate between two multicellular stages, right? Two multicellular stages in their life cycle. And those two are really important for the reproduction of that particular species or that particular plant. And those two stages are gametophyte and the sporophyte. Now, what is the gametophyte? The gametophyte is haploid, right? Meaning it only has a single set of chromosome of its entire genome, right? And that gametophyte produces haploid gametes by mitosis, right? And you have to recall your cell division lectures from your Bio 121, where the cell it's a cell division process that creates a sort of a carbon copy of the original genome, right? The original DNA. And haploid in short term, it's called 1N because it actually creates, haploid refers to having a single set of chromosomes, right? The other opposite is diploid, which is the phenomena where you have two sets of each chromosome in your genome, right? So gametophyte is haploid and it produces gametes or reproductive cells, right, by mitosis. And the fusion of those gametes produced by gametophytes creates a diploid sporophyte. Right now, sporophytes, because it is produced by fusion of gametes, and gametes are haploid, the sporophyte is going to be diploid, right? And the job of the sporophyte is to produce spores, right? And those produced spores are going to be haploid. So if it is haploid, the cell division process is going to be meiosis. And meiosis is a cell division process where your genome is going to be halved, right? So you start with 2N, you end with 1N, right? And by the way, mosses produce spores that actually all look the same, so hence we call them homosporous. Now, the homosporous part is not important for the alteration of generation. What is alteration of generation? That is the phenomena that where you in plants, in all land plants, you observe two different life history stages that are critical for reproduction, right? Gametophyte and sporophyte. Gametophyte is always haploid and produce gametes through mitosis. Sporophyte is always diploid and produce haploid spores through meiosis. Right now, it's extremely important that you remember this definition of alteration of generations, and your lecture will cover it. And if not, you know what that is, right? Now, I keep saying the word land plant, land plant, land plant. Now, there are some aquatic plants, right, that you see in your aquarium. Now, are those not land plants? Well, they are. They are land plants that secondarily moved back to aquatic habitats, right? So all the form of aquatic plants you see that you grow in an aquarium, that you see growing in a pond, that you see growing in wetlands, right, that you see growing in riverbanks, that can tolerate flooding, that grow in floodplains, all of those of plants that are, that actually you see in close association with water, those are land plants that have secondarily moved to aquatic or semi-aquatic or wetland environments, right? Please do not confuse those aquatic plants as non-terrestrial, as non-land plants, right? Land plants, if you go back to my phylogenetic tree, all of those, right, are land plants. And some of those angiosperms, so you see over here, are 100% aquatic. Some of the mosses are 100% aquatic. You might have seen mosses growing underwater. Those are aquatic. But by phylogenetic definitions, all of those are land plants. Okay, I cannot emphasize that any further. All right. Now, alteration of generations, the same phenomena in a diagram, right? You see the sporophyte over here, undergo meiosis, produce haploid spores that produce gametophytes. Gametophytes are now haploid, undergo mitosis, produce haploid gametes, that fertilize, that undergo fertilization, producing a zygote that develops into a sporophyte that is diploid, right? Now, it is extremely important that you use these terms correctly, right? Now, this is something I'm going to emphasize in every day from today onwards, right? So all land plants have a gametophyte and a sporophyte, right? But in a life cycle, in certain plants, right, particularly in bryophytes and ferns, the gametophyte is more prominent in the life cycle. The gametophyte is larger in size. It's conspicuous, right? The sporophyte in bryophytes and ferns are actually smaller in size, right? Less prominent. Very smaller in size, less prominent, and shorter in the lifespan, in their life cycle, right? But if you move to flowering plants, right, and other any form of seed plants, well, it's the opposite, right? The sporophyte is going to be large. Sporophyte is more prominent. Gametophyte is extremely small in size. In the life cycle, the gametophyte has a shorter lifespan. Sporophyte has a longer lifespan. So when you move from very basal plants like bryophytes to more derived, recently evolved plants like flowering plants, right? The prominence, the size, and the lifespan of gametophytes versus sporophytes differ. Now, these names, right, knowing these three phyla is extremely important today, right? And we are providing you the common names, but I do want you to know both the phylum name and the common name, okay? And I'm also providing you the species richness. Don't expect you to memorize it, but I do want you to know that out of these three non-vascular plants, Bryophyta is the most diverse, right? They have over 15,000 species. Okay, let's talk about, yes, shoot, go ahead.
So we're not going to be looking at like specific species of plants, just like the phylum.
So that is actually, we will talk about certain specific species as examples. We have a couple of them on display, but largely when we talk about those non-vascular plants, we are going to characterize the phylum. And just for the display purposes, we will provide you a couple of examples for species. Does that answer your question, Emily?
Yeah, and it says that the phylum goes in the species list, not the species of the phylum.
Oh, I understand your question. You are asking, are you asking me, in other words, what am I going to put into my species list?
Yeah.
Okay, yes. So yes, so what you are going to do in the species list is actually remember the examples I mentioned about? Yeah, so those couple of examples that actually I will actually pick, bring this back again. Yeah, for those examples, you are going to go into the species list, right? Let's say Markentia, like one example for that is actually, Markentia sp, that is the species name, and then that you can go into your species list, right? And then if I don't, and I will, for every phylum, I will at least have a couple of example species. So answering your question, yes, you will have things to go into, actual species that will go into your species list. Does that make sense, Emily?
Yes.
Yeah, very good. Now, non-vascular plants, generally, right, all of them are smaller, right? At least the extent non-vascular plants that are living non-vascular plants are small, right? The reason is because they don't have the vascular tissues, the transportation system to distribute water, minerals, and food around to maintain a larger body. Now, any one of you are, interests or enthusiasts of paleobiology of the planet Earth, right? During the Jurassic period and in Ordovician period, like, at the time land plants evolved around 500 to 475 or 475 to 525 million years ago, there were giant non-vascular plants. There were, right? I mean, those non-vascular plants that were as tall as a rainforest tree. But the climate is different today, right? And there are bigger competitors, right? So because of many reasons, those ginormous morphology of non-vascular plants no longer exist. We don't exactly know why they went extinct. Is it competition against vascular plants? Maybe. Is it the changing climate? Maybe. So we don't know what exact reason actually led to the stunted nature of avascular plants or non-vascular plants, but bottom line, today they all are very small in size. Non-vascular plants do not have true leaves. They have true tissues, by the way, but they don't have true leaves. They look like leaves, but they are not really leaves. They don't have seeds. They produce spores instead. Don't have flowers. They have flower-like structures, but they are not actually flowers, right? And they don't have real roots either. What they have is hair-like extensions, which we call rhizoids, right? Of which the job is to anchor to the most part and subsequently to absorb water and minerals to a certain degree, right? But I must mention those rhizoids are not that equipped, not that efficient at absorbing water and minerals, right? Its primary job is to anchor. If you look at their ecological niche, they largely occur in highly humid, moist-rich, wet environments, particularly like not just humid, but where you actually have some standing water. They're low growing. They don't actually, they could erect, but they don't erect upright that much, right? They sort of have a very low growing, stunted profile. They reproduce, they don't produce seeds, so they reproduce through spore production. Spores are not seeds, and the growth of those spores require a moist, water-rich habitat. And yes, these are the first plants to evolve among all land plants. So first, let's talk about one example, Hepatophyta liverworts, right? Now, they're the closest living plant group to ancestral green algae, right? So the closest land plant relative to ancestral green algae are liverworts. They can reproduce asexually, right? And how they do that is actually, if you look at a liverwort, in fact, you might have seen liverworts, right? Especially those who are nature enthusiasts. Next time you look at a liverwort, like take a very closer look, you will see these kind of really small, tiny cups, right? Sort of literal cups embedded onto the plant, right? Those cups are called gammae, right? And those gammae, if you separate them out from the plant body, they can grow into a completely different plant, right? So those are asexually or vegetatively reproducing units. They normally grow as a thallus, so we call it a thallus. Spelling is like that, okay? And you can call it a thallus growth, right? Meaning what? Look at over here in this picture, in this diagram, right? The thallus is actually growing along the substrate. Here, this is a rock that is a substrate. So this is a plant growing along on the top of the rock, right? Like, it is flat. It doesn't erect upright, right? It grows horizontally along the substrate. And some of them actually can grow along the surface waters, right? And this is actually a close-up of a gammae, right? One of those cups, tiny cups on the plant body. Now, those are, again, not leaves, okay? They are not true leaves. The reason is true leaves should have a vascular system, right? So they don't have a vascular system anywhere in the plants. Hence, we don't call these leaf-like structures true leaves at all. And what you call them is a thallus, right? So they do have this thallus, right? Carry pores for gaseous exchange, right? And this one is actually a microscopic slide of a gammae, right? So gammae is sort of a depression. It's a cup-like depression, right? And then if you look at the underside of the thallus, right, you see the trizoid, right? It's kind of like the entire underside, right? Not just a part of it. The entire underside has those hair-like extensions that helps to anchor the thallus onto the rock, onto the substrate. And some of the thalluses could be like this, like, hair-like, but most of them are like, much more flatter structures like here. Now, the thalluses, some thalluses could actually have female reproductive units. Some thalluses could have male reproductive units. Now, do they have male plants and female plants? No, that's not the case. But what I'm saying is some thalluses, right, of a given plant could produce male units, some female units. The female reproductive units are called archegoniphore or archegonia. Male reproductive units are called female reproductive units are called archegonia. Male reproductive units are called antheridia. And I will show you the spelling later on, right? So bottom line is that liverworts do not have male versus female plants, period, right? And if you look closely, if you look at an actual single plant, you will see both reproductive units on this on two different thallus of the same plant. Now, the next group is hornworts, Anthocerophyta, right? Now, Anthocerophyta, one more thing I want to mention over here is actually, the reproductive units, male and female reproductive units, right, are kind of located on a top of a stalk, right? So this is the stalk, and then this is actually the reproductive unit. This is the stalk. This is the reproductive unit, right? That is something that I want you to know. That's also a characteristic feature for liverworts. Of course, and then again, not having stomata. Instead, they have pores. That's also a really cool characteristic feature. Now to hornworts, they don't have a stalk or a setae, right? That actually sort of holds the reproductive units, right? Instead of a setae or a stalk, right? They actually have sporangium, right? Directly splitting across the length to release the spores to the environment, and which actually subsequently can undergo germination. And then the growth of the thallus happens against the ground. So because of that reason, you can see that in this particular picture, the actual thallus, right, the flattened blade-like thallus grows alongside the substrate, not uprightly erecting from the substrate, right? But the actual sporophyte, interestingly, can erect at least partly off the thallus, right? And these guys actually have not just spores, actual stomata. And this is an actual stomata taken from a hornwort, right? But those stomata are not located in the thallus, right? Not in the gametophyte, but it is located in the sporophyte, right? And that stomata actually can open and close as needed. Now let's move to the third group, bryophytes, mosses. Again, the most diverse group of land plants, most diverse group of non-vascular land plants. Now they also have a setae, a stalk that holds the sporangium on the top. Like this is the setae or the stalk, right? And this is the sporangium that actually contains the spores. And the sporangium is sort of high up in the air. There's a reason for that, right? The reason is actually when it is high up in the air, it actually can easily get dislodged by wind and disperse somewhere else. As opposed to over here, the sporangium, right, is actually not that high up in the air, and it's sort of more longer, right, with a very short stalk, if there's a stalk at all, very short stalk. So you have a longer sporangium which splits across the length, but over here you actually have a much more shorter sporangium but a longer stalk or a setae, right, which actually is better exposed to air, right? Now these mosses too, they actually have stomata on sporophyte, not on the gametophyte, right? So the same thing, it's a characteristic feature they share with hornworts, right? Now these guys actually have separate male and female plants, right? And so because of that, and then they produce spores, and all the spores they produce from the sporangium are actually similar. So hence we call that homoporous. I will explain this particular plant part subsequently, but I intentionally put it here that you can understand where these parts are, right? And I will explain the anatomy in a subsequent slide with respect to your exercises. And let's go over the pictures over here, make sure I haven't forgotten anything. Yeah, over here, right? So this is actually moss, which you have seen growing all over rocks and sometimes on logs, particularly on like stumps or like right at the root of a tree trunk. And that is like what you mostly observe over here is the gametophyte, right? And the sporophyte is actually this part that erects from the gametophyte, right? So same thing over here, right? The sporophyte is this greener part. The much more darker, browner colored part is the actual gametophyte, right? So the thallus part, right, over here, right, is actually the gametophyte, right? Now, moving over here, right, let's talk a little bit more about the moss gametophyte in detail, okay? Now, the spore, right, released by the sporangium, right, sporangium singular, sporangia plural, the spore that is released from the sporangium germinates first into a gametophyte, right? And remember, spores are produced by a sporophyte, right? And in the sporangium through meiosis, so those spores are going to be haploid, right? And because of that, the gametophyte is also going to be haploid. And that gametophyte, has two parts in it: the protonema and the gametophore. Protonema and protonema is actually sort of the branching part, and the gamete producing part is actually called the gametophore. And I will show you a diagram in the exercise to make that a little bit clear. And the gametophyte is a multicellular structure, and the gametophyte, particularly in the gametophore component, produces multicellular sex organs, and those multicellular sex organs are called gametangia. I know there's a lot of terms, so I will keep repeating the terms and showing you those terms in diagrams to make it clear. If it is not clear, if it is confusing, please keep asking. And the female gametangia is called archegonia. The male gametangia is called antheridia. At maturity, right, each archegonia, the female part, right, contains a single egg, right? Single egg only. Antheridia, on the other hand, the male component, the male reproductive organ, has many sperms and produce many sperms, right? Like I mentioned earlier, for most gametophytes, the height is constrained by lack of vascular tissues. Without vascular tissues, you cannot maintain a taller tree, right? So the gametophyte, although it is prominent in the life cycle, it is still short, quite short and smaller in size. Rhizoids help to anchor the gametophyte to the substrate, to the soil, to the rock, or to the log that the moss is growing on. Mature gametophytes produce flagellated sperms. Remember the male reproductive parts, antheridia, they produce these flagellated sperms, right, in the antheridia, and the egg is produced in the archegonium. And what the egg that is produced in the archegonium remains in the archegonium forever, right? They don't leave. So for fertilization, what happens is sperms have to actually leave the antheridia, swims through water, and enter the archegonium, and then fertilize the egg inside the archegonium. So if you think about it, this is sort of an internal fertilization, right? Because the fertilization happens inside the female organ of the plant, right? So again, just to highlight, the thallus-like part that is growing, along the substrate that is flatter, right, blade-like, is the actual thallus is the gametophyte. This particular part that is somewhat upright, that looks like a tiny plant, tiny palm tree, right, is the gametophyte, right? Okay. sorry, is the sporophyte, right? This is the gametophyte, the thallus-like part that is spreading on the substrate, that is blade-like, leaf-like, is the gametophyte. The upright part, right, is the sporophyte, right? And clearly the upright part is much smaller, right, and less prominent than the thallus-like part. And again, these tiny structures, those are the cups, the gametes, the gamete that helps asexual reproduction. All right. Now what is, I mentioned two terms over here, gametophore and protonema. Okay, what is protonema? Let's tackle that first. Protonema is sort of the initial growth from the spore, right? Like I mentioned, when the spores are released by the sporophyte, right, or produced by the sporophytes and released by the sporangium, those spores will start germinating, right? So the initial growth that starts, right, from the spore is actually protonema, right? And the protonema, well, will actually continue to grow, continue to branch, and producing more extravagant, more extensive biomass. And then the protonema will subsequently produce the gametophore, and the gametophore is actually responsible for producing reproductive structures, right? So in these diagrams, right, all of these green structures you see that are filled with chlorophyll, that are green in color, right, that is the protonema. That's kind of the mycelium of a fungus, right? Sort of an analog, right? And then over here, this particular part that actually looks like a plant that is growing upright is the gametophyte. That is produced by gametophore, right? Now, like I mentioned, protonema is growing straight from the germinating spore, right? In most mosses, an extensive branch system of multicellular filaments that are rich in chlorophyll. So like I just pointed out, the protonema is extensive, it's multicellular, looks like a network, a filamentous network, sort of analogous to mycelium of a fungus. Initiates the accumulation of hormones that influence the further growth of the newly formed cells. What does that mean? The job of the protonema is actually to permit, to allow, first synthesize hormones, secondarily accumulate hormone in a certain threshold concentration that will trigger a further growth of the protonema itself, to the growth of the gametophore and subsequently the gametophyte. When specific concentrations of the hormones are reached, the branches of the protonema generate small buds, which then turn into a gametophore. Yes, so what happens is, yes, first, the hormone concentrating hormone levels will lead to extensive development of, further development of the protonema, and further accumulation of the hormones will actually lead to development of the gametophore. So, now to the sporophyte. That was about the gametophyte. Now let's talk about the sporophyte. Bryophyte sporophytes grow out of the archegonia, right? So how come? Because remember, right, archegonia is where fertilization takes place. Archegonia produces the egg, the sperm swims into the archegonia, fertilizes the egg, makes the zygote, and zygote grows into the sporophyte, which is diploid, right? And in mosses, in any vascular, non-vascular plant for that matter, the sporophyte is very small, much smaller than the gametophyte, right? And the sporophyte consists of a foot, and the foot simply anchors the sporophyte onto the gametophyte, right? And a setae or a stalk, right? And the setae terminates in a sporangium, right? And also known as the capsule. If sporangium is too big of a word, use capsule. And that capsule contains spores, which upon rupture can discharge spores to the atmosphere. Now, releasing spores from a sporangium can be controlled. The peristome, right, which sort of means like a mouth-like organ, its job is to regulate the release of sperms. So because of the peristome, a particular sporophyte does not release all spores in the sporangium. Rather, it actually releases a tiny bit at a time. And in fact, hornwort and moss sporophytes have stomata for gaseous exchange. Liverworts do not have stomata anywhere, right? None of the gametophytes have stomata, right? It's only hornwort and moss sporophytes that have stomata, right? Liverworts have no stomata at all. Instead, they have pores, right? And now let's again revise this moss anatomy, because there's a lot of words that I want you to understand, right? So let's look at this diagrammatic picture first, right? So sort of the below ground part is the rhizoid, right? The rhizoid helps to anchor the entire plant to the substrate, right? And sort of extends vertically into the substrate, to the soil, or sometimes even to the rock, right? And they are pretty strong structures. Then protonema is the first development, the initial development of the plant from the spore, right? This is protonema. And then the structures that arise from protonema, right, is the gametophore, right? This part is the gametophore, right? And it has this leaf-like structures. We call them, we have a particular name for them, and I will introduce the name later on. And then what happens is at the end of the gametophyte, right? If it is a, depending on what kind of a plant phylum it is, right? If it is in case of mosses, like in male versus female plants, in a male plant, at the end of the gametophyte, you have antheridia for female gametophytes. At the end of the gametophytes, you have archegonia, right? So in the female plant, the archegonia has an egg, and the sperms produced by antheridia swims into that and fertilizes the egg, right? And after fertilization, a zygote is produced, and the zygote then develops into this particular part, which is the sporophyte. Again, quite literally, sporophyte is located on the female gametophyte, right? And then the parts of that sporophyte, the foot part, which helps to anchor the sporophyte onto the gametophyte, right? And then the long stalk, or the setae, at the end of which you have the sporangium that produce spores. This entire structure, right, is called the gametophyte, right? So gametophyte equals gametophore plus protonema plus rhizoids, right? And sporophyte is located on top of the female gametophyte, or the female part of the gametophyte, right, which has a foot, setae, or a stalk, and then the sporangium, or the capsule. Okay, and then in a real life, right, sporangium is on the top, like just like that, and then you have the stalk, and then the foot, and collectively all of these are the sporophyte. Where do you find this sporophyte? Always on the top of a gametophyte, right? And then I'm answering a question from Emily. So I'm introducing you an example species for the first time, actually, genus Sphagnum. So Sphagnum sp, right, is actually, something you can put into your species list, right? And its common name is actually Sphagnum moss. It's really weird. The scientific name and the common name is actually the same, right? It's a really cool plant that is growing on peat. It's actually an indicator of healthy wetland environments. Okay, that's actually end of the introduction. Any questions, guys? Okie dokie. Now we are actually getting into more interactive part. So I ask you guys to actually, be interactive and open your videos back. That actually helps us to, carry on these conversations more effectively. So I, please urge you to, and thank those who are already turning with their videos on. Can everybody see? Give me a nod or a shout out. Can you see the new PowerPoint? Thank you. Moving on. So first, I have actually uploaded really two cool short videos that explains the reproductive parts of that. And I will explain it myself, but I kind of hope that, you know, the other two videos will help you to learn the same material. It's extremely important for your lecture as well as lab. First exercise is actually the key, right? Let's do one example. So this is the very first time you are doing plants, so I will give you at least one example. Then if you need, I will do a second example. Does that sound good, guys? Alrighty. So very first one, read with me. Gametophyte thallus, right? It's flattened in appearance, living on soil. Gametophyte is foliose in appearance, living on soil or in water, or forming a mat of green thread-like structures, growing on agar. Right, let's look at the number one. Okay, take a good look at that, right? And it says plants are growing flat against the ground, and you can see it's actually, you know, it's sort of growing against a log, and it is growing like literally alongside the log, right? Take a look at that, and it's green in color, green, yellowish green in color, right? And then let's go back over here, right? Now, tell me, guys, this is the interactive part. Give me my pointer, please. Here we go. What do you think? 2 or 3? Is it 1A or 1B, guys?
1A.
Okay, 1A. Yeah. Well,
I was gonna
now,
say 1B because it looks leafy.
yeah. So let me go back and explain you what thallusy or thallus-like means, what leaf-like means. Literally over here, you can actually see tiny, tiny, tiny leaves in these things, right? But if you look at over here, that is what a thallus looks like, right? Flat blade-like structures, you know. And over here, so we will actually go to B, right? And let's go to that's going to be B1B, that is going to part 3. Gametophyte aquatic, gametophyte terrestrial or growing on agar, right? So gametophyte aquatic in water, gametophyte terrestrial or growing in terrestrial like environment, agar, right? Or auger. I don't know how you pronounce it. Do you guys know what auger is?
Sort
Auger?
That's just artificial or whatever thing that you have on petri dishes to grow mold or bacteria or whatever it is.
Correct. It's an artificial growth medium which actually provides nourishment. It's largely a carbohydrate based growth medium, right? That is 100% artificial, right? It simulates terrestrial or hard substrate environments that facilitates growth of things ranging from bacteria, fungi, even to these to any form of land plants, right? You can actually grow a tiny seedling on agar. Okay, auger. Doesn't matter how you pronounce it. Now, let's go back to over here. So what do you guys think over looking at this? You know, it's clearly growing on the ground, not aquatic, right? So in point 3, what are we going? 3A or 3B? Clearly.
3B.
3B, very good. Let's go to 3B. It says go to point 5. Point 5, gametophore, the reproductive structure of the gametophyte, growing flat against the ground with obvious phyllids or phyllids lacking. Now, phyllids is something I didn't introduce you to you yet. Those are the leaf-like structures that you see in plants, right? So obvious phyllids or phyllids lacking, green in color, thread-like in appearance, right? And forms a mat. That's one option, right? There's a long option, right? If either phyllids present or phyllids absent, right? Green in color, thread-like. Other option is gametophore upright with radially arranged phyllids. Where would you go with that? 6 or 7? Or 5A or 5B?
5A.
5A. Others agree? Okie dokie. 5A. So that goes to 6. Gametophore flat against the ground with obvious phyllids. Mat of Protonema onega. Well, actually, the ega part doesn't apply, right? So it actually goes to this one, right? All right. Do you want me to go to another one, or are you all set to do the rest by yourself?
Do another one.
Okay, I'll do another one. What are we going to pick? Let's pick the number 2 one, right? This, right? Look at that. Take a good look at that. You know, it actually has this green color. It's, you know, has a thallus-like appearance, light greener in color, thallus-like in its shape, right? And look at this, right? It actually has this bifurcation, right? It divides into two parts. The thallus keeps dividing into 2 parts, right? Like you can see it over here very clearly, over here very clearly, and over here, right? It's dividing into 2, right? And again, you can see first it branched into 2 over here, right? And then each part again divides from here into 2, right? It keeps dividing into 2s, right? 2 parts, 2 parts, 2 parts. That's called bifurcation. Now, knowing that is important to complete this exercise. 1A or 1B?
A.
Yeah, clearly, right? You go to A. That is actually 2. Dichotomously branching, meaning branching into 2, bifurcating, right? Thalluses with elongated sections, yellow-green in color, thallus lacking a cup-like structure. Let's go back and see whether they actually have cup-like structures. Nope, I don't see one, right? I don't see cup-like structures, right? So what is your choice? Thallus, the other option, thallus rounded, olive green, cup-like structures present. So clearly, yep, we are looking at 2A option, right? Okay, there we go. We did 2 of them. Are we good? Alrighty, very good, excellent. So let's actually skip through all those examples. You know, look at this one. This is actually, you know, another good. Come on. Another good example for those gamma, the cup-like structures, right? And here, look at this. These are actually more round thallus, right? They don't actually necessarily keep repeatedly bifurcating, right? And that's actually an example for agar-based growth. Now to the mossel's life cycle, right? So I will quickly go over this life cycle. Now remember, life cycles are complete. But before I go over, I'm going to ask you, are you okay with following this life cycle yourself, or shall I explain in my own way? Completely up to you guys, because I'm asking that question because I know you cover this part in lecture 2.
I feel pretty confident reading it myself.
Okay, others?
I wouldn't mind an explanation.
No, I don't mind at all. Even if just one student mentions, I'll be quick, right? So now I have personal preferences to start a particular life cycle. Like I usually start with the spore, right? But remember, it's a cycle, right? You can start from anywhere that you are comfortable with, right? I'll start with the spore, and spores, remember, they are haploid. It, you know, has a single set of chromosomes. They keep growing, first producing the protonema, right? And then the protonema growth keeps continuing to the bud formation stage, right? And then the protonema completely, completes its development, producing this male gametophyte, right? Remember, I'm talking about mosses, so now I can tell male gametophyte, female gametophyte, right? So, and then again, another spore generates a female gametophyte. It's interesting, the spores don't look different. They are homoporous, but some spores can produce male, some produce females, right? So, and then when you have a mature gametophyte, male and female, it has a rhizoid, it has this, the gametophore and the rhizoid component, right? And collectively, that is the gametophyte, right? So the male gametophytes produce archegonia right on the top over here, archegonia. It's a reproductive unit. Archegonia actually has an egg, right? We are talking about the female gametophyte, right? And that egg stays inside the archegonia, right? It does not leave the archegonia. Now remember, Remember this very carefully, right? That female gametophyte has multiple archegonia, right? But in each archegonia, you only have a single egg, right? So this is an archegonia. This is an archegonia. This is an archegonia, right? So each archegonia has a single egg. Let's look at the male gametophyte, right? Male gametophyte has this gametophore part and then the rhizoid part. On the top has multiple antheridia, each antheridia producing so many sperms, and all of those sperms are released to the environment and literally swims through water, right? So remember, for antheridia to be released, for the fertilization to happen, they require water. Without that, they cannot. So in a dry environment, mosses cannot reproduce, right? They swim through water, right? Enters the antheridia, enters the archegonia, fertilizes the egg inside the archegonia, right? And remember, archegonia is on the top of the female gametophyte. And then the fertilization results in a zygote. Again, think yourself, you have a haploid sperm, haploid egg, right? Why are they haploid? Because they both are coming from a haploid gametophyte through mitosis, right? They fertilize, producing a zygote. Zygote after fertilization, logic should tell you it's diploid, right? And that diploid zygote is unicellular at first, immediately undergoes cell division, become multicellular, right? Mitotic cell division and become multicellular. And that multicellular embryo develops, grows into a diploid sporophyte. Again, you don't have to remember every step, right? I mean, the logic comes in now. If the zygote is diploid, the sporophyte must be diploid. And again, start thinking, okay, where is a sporophyte? Is a sporophyte going to be on land, in water? Nope, it's still inside, happening inside the archegonia. So it's on top of the female gametophyte, and then ultimately develops into a mature sporophyte. Mature sporophyte has a foot, stalk or the setae, and the capsule or the sporangium. What is happening inside the sporangium? Meiosis to produce haploid spores, right? And then again, remember, this spore release happens through the peristome, and the peristome is actually a structure that regulates how much spores are released from the sporophyte or from the sporangium, right? Remember, sporangium is the part in which spore production happens, right? And sporangium is a part of the mature sporophyte, right? And there's only a single mature sporophyte for a single, there's only a single sporangium for a single mature sporophyte, right? Remember that. That's as simple as the life cycle, right? So here, this is for you. Like, first go through this, go through my label diagram. Really? Go through my label diagram over here and then label it yourself, right? This is sort of a practice. Now, this is again the same, but what I have done is, instead of using, like pretty pictures, I'm using slides, right? And the next part of the PowerPoint of the exercise is actually to in detail understand each and every unit in this particular sort of synthetic life cycle, right? So sort of a recap of all the terminology that I want you to know, what it is, when it occurs in the life cycle, and you should be able to identify it if I show it to you through a slide or a picture or a photograph. And the link over here is the same link that I showed you earlier. Okay, it's not a different link. So first, we will look at the archegonia, right? The female reproductive structure that you locate on the top of the gametophyte. So in a slide, it looks like this, right? There are multiple archegonia on the top of a single gametophyte, right? And then if you zoom into that a little bit, it looks like this, right? And inside of that, you find this tiny little egg, right? This is the archegonium. There are many archegonia, one over here, there's one tiny one over here, right? And over here, there are 1, 2, 3, 4, 5, 6, I cannot even count. So many. Like in each archegonia, remember there's only one egg. Now let's try to label this. Help me out here, right? So what do I call this entire structure on the top of the gametophyte? Right, that sort of is holding all the reproductive units. It ends with 4.
Gametophore?
Correct. That is gametophore. Now, what do you call this particular structure? The cell, the cellular part that contained, sorry, not the cellular part, sorry. The circle structure, the female structure, the red waist-like part that is inside the red circle. This part. What is that?
I can't think of how to say the word, but it's what you just went over on the last slide. It holds the egg in the female.
Yeah, start with the letter A, right? Archegonium or archegonia, right? Guys, I know the words are like, yeah, as an ecologist, animal biologist, yeah, I understand the words are extremely painful, but we get to used to it. What do you find inside the single cell that you see inside of it is
The egg.
correct. After being fertilized, the egg becomes
Zygote.
correct. Excellent. Now sporophyte. This is actually how a photograph looks like. Then again, answering Emily's question, this is actually our example, right? Now, when you just observe a moss, right, in the environment, when you're hiking, right outside your home, what you see is actually the gametophyte, right? Because gametophyte is larger, more pronounced in the life cycle and lived longer in the life cycle. But take a closer look, you will see this, the brown colored thread-like structures, and you will wonder what the hell are these, right? Take a very closer look. When they are ready to reproduce, when they are more mature, you see both the green structure on the bottom, that is the actual gametophyte, and the brown non-green structure, the sporophyte, on the top. Check more closer. It looks like this, right? And it looks like sort of a seed coming up or a flower coming up. Not a flower, not a seed. It is actually the sporophyte, right? So over here, what you see is actually on the bottom, right? This greener mass is actually the gametophyte. These things that is sort of popping up are the sporophytes, right? And the sporophytes are not anchored to the substrate, rather they're anchored onto the gametophyte. I will keep repeating this just so you absorb it, right? And also, this is not something I think this is additional, right? Look at the color of the plants. Do you think the sporophyte can photosynthesize? Mostly, the sporophyte is not photosynthetic. And then in some literature, I read that they use even the term the sporophyte is parasitic on the gametophyte, right? Just keep that in your mind. I'm not going to test you on that, because certain plant biologists will debate on that. So again, let's label this. This brownish part on the top is what? That produces spores?
Sporophyte.
Correct. And the bottom part is?
Gametophyte.
Gametophyte. Right, guys, I do want all of you to actively take part, right? This is supposed to be collective. So I'm not going to go over this with you guys, because I showed the same image earlier, right, in my introductory PowerPoint. So I will let you label yourself. Again, sort of relabeling this over here, right? It's what is the bottom part, what is the middle's longer thread like part, and what is on the top of the top, right? You can easily go back to my PowerPoint presentation and learn what that is. Now let's talk about the sporophyte in detail, or particularly sporangium in detail. Sporangium is on the sporophyte. Now, how do you remember these things? I can give you sort of the etiology. Gameto, like phyte is the plant. right? Phyte means a plant. Gametophyte, gamete producing plant, gametophyte. Sporophyte, spore producing phyte, spore producing plant, sporophyte, right? So that's actually one easy way to remember. Sporangium, right? A capsule that contains spore, sporangium, so like that. Breaking down the Latin term might help you to understand it. So again, what you're looking at over here, the entire structure is a sporangium, right? It has a stalk or the setae, right? Then the actual capsular part, right? So the top of the capsule, when it is first developing, has an operculum, sort of a cap protecting the internal developing spores. When the sporangium matures, the capsule disintegrates, making the peristome, right? That actually regulates the spore release. The shape of, so this is actually what your example that you would see if you were meeting in person in the lab, right? So the spores are the red dots inside, right? And then there are some other mosses like the sphagnum moss, has a completely different shape, right? It's more like a oval over here, right? Again, the same thing, right? The stalk, right? The actual capsular part, right? And the spores located inside of it. Again, you can easily label this yourself, right? Now, label this part with me. Let's do that since we skipped several other things, right? In this particular diagram, right? What would you call this green colored, right? Elongated structure. What do you call that? It is green. It's a filament like, it keeps branching.
Seda?
Sorry? Setae, you said?
Yeah.
No, let's start actually, probably that's harder question. Let's start with this one, right? Structures that used to anchor the plant. This thin hair-like structures that is extending all over, that are light green in color. What do you call those? That the purpose is anchoring. Those are?
The rhizoids?
Yes, rhizoid, correct. Now, if that is the rhizoid, what would you call the more thicker green-like structure? There are potentially two correct answers over here.
It's like the fungi mycelium, but I'm blanking on the name.
Yep. So you can call it either protonema, that wouldn't be wrong, or you can call it gametophore, right? And then last one, cells produced by the sporophyte. What do you call the things that are released by the sporophyte? That's simple. Yeah, Randolph?
Aren't they haploid cells?
Yes, they are haploid, but what's the name?
The gametophytes?
No,
They turn
gametophyte.
into gametophytes, right?
Correct. It develops into a gametophyte. Like what does the sporophytes release? Sporophyte is a tree that produces?
Spores?
Yes. So the cells produced by the sporophyte is actually going to be spores, right? Okie dokie. And then again, sort of a close-up example of another example, moss, right? Labeling exercise, right? We actually talked about most of this already. So remember, I mentioned what you call in our key, right? What do you call these leaf-like structures? Starting with the letter P. Phyllids, right? The leaf, quote unquote, leaf-like structures. Those are the phyllids. The other parts, actually, we already labeled them several times. What are we looking at over here? We are over here looking at the another reproductive structure, right? Another gamete producing structure. So let's try to label this together, right? So the whole structure, the upright structure over here that is producing gametes, and provided that it is producing male gametes, right? What should that be? What should this entire structure should be? Go for it, guys.
The antheridia.
Now, antheridia is located inside of that. So what structure holds all the antheridia together? It ends with 4 and it is producing gametes. So gametophore, male gametophore, right? Do not forget to say male, female. It's really important. And now these things that is located on top of the male gametophore. Randolph, you already gave me the answer. What is that?
Antheridia.
Correct, antheridia. And the antheridia is going to produce what? What is the male reproductive cells are called?
Sperm cells.
Correct, sperms. Correct. Again, we actually did label this part already, so I am skipping this and going to the exercise 3. So again, like I just to not to like give you a pain, but when you are doing the exercise yourself, I'm adding different photograph just so that you understand like what these structures are, right? What is producing, what structure produces spores, what structure contains spores, right? Now let's take a look at the cuticle, right? Now if you take a phyllid, right, a piece of the thallus of the gametophyte, right, and observe it under the microscope, right? First, you are going to see lots of cells, right? In between cells are these yellowish colored things, and then you clearly see the cells are not tightly packed with each other, right? Something covers all the cells and surrounds the entire structure. Now this red margin that you see over here, right? That part is actually the cuticle, right? And then if you just for the comparison's sake, right? Now we are looking at a fern, right? Right on the top of the phyllid, right? Or the leaflet. You see a really thin, transparent structure that protects the cellular components, right? That is also the cuticle. Now you find the same if you look at a vascular plant on the top of the leaf blade, a non-cellular component that is transparent, protecting the entire leaf. That's also the cuticle. So cuticle is observed in a lot of lineages of land plants, right? And those land plants and those cuticles are protective in function. What I need you to know over here is that the moss or the non-vascular plant cuticle is extremely thin. It's thicker in the ferns. It's even thicker in vascular plants, right? That's something I want you to learn, right? So maybe presence of a cuticle is not unique to bryophytes. Maybe it's not unique to mosses. It's not unique to all land plants. Oh, well, it is unique to all land plants. It's not unique to all non-vascular plants. But if you want to, mention something unique about the cuticle of non-vascular plants, you can mention they have the thinnest cuticle of all land plants. Pores, right? Now, if you look at this, there are two examples over here, right? If you're looking at the leaf blade, if you closely look at the leaf blade, right, maybe with a, dissecting microscope, right? Here, look at the multiplication, the magnification, it is about 35 times magnified. You will clearly see individual cells in the center of each cell on the thallus. you will see an opening, right? Now that is actually the pore, right? Now, if you are looking at a liverwort, it's just a pore. But if you are looking at a moss, that is definitely a stomata, right? If you're looking at a hornwort, you are looking at a stomata, right? And again, additional pictures for display, right? What a moss sporophyte looks like, right? And what a hornwort gametophyte looks like, right? So hornwort gametophyte is over here. And this long structure that is spreading from that is actually the sporophyte. And this particular part, you can see it is turning brown because it's actually producing spores, right? And over here in a moss, this is the moss gametophyte and this is the moss sporophyte. And another thing I want to mention to you is actually, I told you that when it comes to mosses and hornworts, right, the actual, there was a chat. I did mention that the stomata in hornworts and mosses are only limited to the sporophyte, right? But the spores, the actual pores can be produced, P-O-R-E-S. Pores can occur in the entire gametophyte, right? And when it goes again back to liverworts, they only have pores, right? Alrighty. Now there are several displays, right? Actually, well, not several displays, just one display, the background that you see, right? And the questions that I expect you to answer in your lab notebook is, name 2 advantage of mosses forming a thick mat with respect to water retention, right? Well, think of what a sponge does, right? When you produce a mat, I mean, go and touch a mass of moss. It looks like, it feels like a sponge. So it sort of collectively, the gametophyte itself, with some help from the rhizoids too, retain water, thereby actually increases the humidity of that environment. And then and prevents the plant from drying out. Number 2, what type of organisms might you find living underneath and in these thick masses of these thick mats of mosses? Well, we just finished talking about animals, right? And we mentioned that so many animals are actually dependent on moisture, right? Well, insects are rather independent of moisture, but there are several even arthropods that are moisture dependent. There are several annelids that are moisture dependent, right? There are certain free-living flatworms that are moisture dependent. So which one could be actually, which kind of ones are those, which one of animals are, moisture dependent that can easily get their skin dry, right? If you can think about one, you will expect them to live over here. Now, don't forget to think about vertebrates as well here, right? When I taught vertebrates, I mentioned several examples, right? Particularly amphibians that actually are dependent on moisture. Well, some of them actually, particularly salamanders, lungless salamanders in particular, can actually occur underneath the mosses. And actually, there's a particular species that I study, right, that only breeds, lays eggs underneath the mat of mosses, right? So there are really cool things you can talk about there. Question number 3, based on the organisms that you just listed in question number 2, what other organisms would be attracted to these mosses? Now that question is, of course, is about like, okay, now provided that there are certain animals that are attracted to moist, wet, humid environments, what other animals will come there just to eat the others? So the question is as simple as that. And that is the end of the exercises. So the last part today is to go over not that, go over this. Ouch. Go over the homework. Any questions so far regarding the exercise? Anything that you guys want me to clarify? Very good. let's look at the homework, and I think you can all see the homework as I do. And then, by the way, if you might have noticed that I have gone faster, yeah, because you are getting used to this, you are getting more comfortable with what is expected. And it's mostly, being accustomed to the plant components, the plant parts yourself, right? So if you need me to explain something back, please don't hesitate to ask, right? And then I know there are students who will appreciate if you ask questions, and I do, right? Appreciate if you ask questions. Okay, let's go with the homework part. The first part is actually complete the table using your notes from your life cycle, exercise, and observations. Right? If you have not done so already, record this information in your lab notebook. So scientific name of 2 species from the PowerPoint, like from the presentation we just did, right? I mean, I have several examples, right? So the charophyte example, the green algae, like I'm providing it already for you over here. You don't have to provide another, right? The other one, the only thing you have to provide is provide 2 examples for mosses, right? I do have several in my PowerPoint. Just go look it up, right? And then what is a phylum? Well, for the charophyte, it's Charophyta, right? For the mosses, it is Bryophyta. And then structures where the photosynthesis happens, well, the green leafy structures, right, on the mosses where the photosynthesis happens, that is called phylids, right? Don't say leaves, don't say even leaf-like, right? Those are called phylids. Another answer is thallus. Thallus itself is green, right? Now, if you write gametophore or gametophyte, I'm only giving you partial credits because you can be more specific. Okay, or if you write a protostome, protonema, I will only give you partial credits. Phylids or thallus, I will accept as full credit answers. Structures that is used for anchoring, right? What do we call that? Can someone remember? Can someone tell me? I use the word a lot. Emily?
Rhizoids.
Correct, Emily, correct. And structures for gaseous exchange, there are 2 names that you can use. What are those?
Pores.
Good answer, right? Pores, right? And another one starts with letter S.
Stomata.
Correct. Now, when you say stomata, specifically mention what part or what life history stage contains stomata, right? We are talking about mosses, so you have to be specifically mention stomata of the sporophyte. That way, actually, when the exam comes, you will not forget these things. Structures for water uptake. So I did mention earlier that the rhizoid can do that for a particular part. What I didn't mention is that the other parts of the protonema, right? The other parts of the gametophyte, the gametophore itself, the thallus, the phylids, they themselves, to a very, very limited degree, can absorb, right? Particularly when the cuticle is thin, they can absorb water through diffusion, right? And also the pores, right? They will also directly take up water, right? Not just gaseous exchange. So structures to protect from desiccation. Shout out an answer. Everybody, guys, I do see a lot of answers coming from Kayla, Emily, and Randolph. Others? All right, anybody, come on. I only have the few continuous participants. What protects from desiccation?
Is that the cuticle?
Thank you, Matt. Cuticle, correct. And the last part, the habitat, right? So habitat is actually where you find it, right? Oh, sorry, not habitat, habit, not habitat, habit. Habit is actually the form of the plant, right? So How do you describe the form or the structure of the plant? Anyone? It's okay to make a mistake, right? I'm not going to pick. How do you describe the plant's shape, structure? Yeah, well, Jared, go ahead.
I was going to say, basically like it did on the dichotomous key earlier, like either describing the parts as flattened or elongated and matted or anything like that.
Correct. So there could be largely, most of them are going to be thallus-like, right? So you can mention thallus-like or flattened, like you just mentioned, Jared. And then some parts are upright, like or partly upright, like the sporophyte component, right? And then the last thing you should not forget is it's short, right? They are not growing tall. Alrighty, moving on to the next part is actually compare and contrast sperm and pores of non-vascular plants. Be sure to include both differences and similarities lists. Ploidity, right, meaning whether they are diploid or haploid, right? The cell division process needed to, what kind of cell division you need to produce that, meaning is it meiosis or mitosis? And then dispersal mechanism, right? And at least one other similarity and one more difference. So dispersal mechanism, meaning is it wind dispersed, right? Remember, spores were wind dispersed, and remember that sperms were water dispersed, right? So like that, what is the dispersal mechanism, right? And then a difference between them, right? So that actually, if you go back to the PowerPoint presentation, you can easily grasp the answers to that. So I will not actually try to call answers right now. The next part is actually really fun because we have provided you a description about a particular plant. We provide its characteristics, right? It says two distinct spots, one green, one yellow. Immediately you should see green part should be the gametophyte. The brown part, yellow part, should be the sporophyte, right? And says green part has radial symmetry. That tells you about the arrangement of the phylates, right? When you touch the yellow-brown part, meaning when you're touching the sporophyte, dusty substance is released. That actually should tell you, okay, that brown part is already releasing spores. And the specimen you take back to the lab, it gets dried out. So it also tells you, okay, they must have either no cuticle or very thin cuticle, right? Very dependent on wet environments. So based on that, although those characteristics, right? You can describe the parts, the green part, like and you can describe like what is the two distinct spots, right? And then B, what specifically is the green part? And what are these leaf-like structures that is coming out of the green part, right? And then what are those structures that is coming out of the brown part, right? And then and the specimen dried out. Why? Well, could be several explanations. You could talk about the thinner cuticle. You can talk about the fact that those are early plants, right? They are non-vascular, right? Early land plants. They are not fully adapted to terrestrial mode of life, and hence they are highly dependent on presence of water. So that's why it dried out. And then, well, we covered 3 different phyla today. Out of these 3, which one do you think it is? And then you can actually provide an explanation. Why, right? Honestly, it could be either of the 3, right? The correctness of your answer depends on how you explain it, right? Why do you think it's a bryophyte as opposed to the other 2, right? So the last part is actually also a much more creative part, and I'm not going to explain that, insulting your creativity. So come up with a new scientific name. right? Try to be meaningful, right? With respect to your scientific name. You did similar activities before, right? And also a new command name. Again, try to be creative with the command name with respect to its habit, its habitat, its form and function, right? And then, of course, you try to modify that particular plant, by adding a particular feature, right? And then, of course, answer those parts of that question. So this part is largely the bottom part of the page 2 is largely testing your creativity, right? Well, that's basically it about everything. Emily, go ahead.
I had a question about the last question.
Yeah. Let me share back that. Yeah, Emily, go ahead, ask your question.
So it's like we have to give it a quality that we think would place it into a different phylum than we originally think it goes in.
That is correct. Describe a change. Yeah, let's read through it since you asked me. The last part, right? Very last part, Emily, right?
Yes.
Describe a change in one feature listed above that would place it into a different non-vascular phylum. Yes, so there are 3 phyla. You picked one earlier based on a particular explanation. And then what is a feature and a change? And is in what non-vascular phylum could it be placed, right? So what kind of a change, right, that would make you change your mind? For example, let's say you observe that this particular earlier you mentioned it's a bryophyte, right? And then now if you want to change it, right, maybe you observed, or if you were to observe presence of actual stomata on the sporophyte, well, then okay, then it is not a bryophyte. Then it is not. Earlier you put it in, my bad. Earlier you put it in the liverwort category, right? Hepatophytes, and then subsequently you observe that they have stomata on the sporophyte. Now you know immediately it's not a hepatophyte, not a liverwort. So it could be either of the other two, right? That's actually one example. Did that answer your question, Emily?
Yeah, I think so.
Yeah. So in other words, it's what kind of a character, what kind of an observation, right? A new observation, right? That would make you change your mind, right? It's not something listed already over there. No, you have to be creative, right? It's not something that is already listed in the top of the page. It's a new feature, right? Hypothetically, if you were to observe this particular feature, right, what would you call it in terms of phylum? Right, that's what the question is. So it's your job to come up with what feature that might change your mind, and changing in your mind from earlier from A to B, right? So you have to decide what change, what new feature, right? That's and then with that changes, what kind of a phylum would you assign it to? That is the question, right? And it has to be a non-vascular phylum, right? Yeah, that's what we are looking for. Is it clear, Emily? If not, I can actually, I will try to explain it one more time.
I'm just a little bit unsure, like how a change in one thing would mean that it would go into a different phyla.
Yeah, so I think the reason the confusion is you're asking yourself, scientifically speaking, does change of a one feature completely shifts its membership? I think that's what we are confused with, right?
Yeah.
Yeah. Well, in that case, you have to pick a particular character that completely sets it apart. For example, let's go back to the example that I gave you guys, right? If earlier you considered this mystery example as a liverwort, a hepatophyte, right? What kind of a change in the characteristics, what kind of additional feature in the characteristics would change its phyla? Well, let's say that hypothetically, this particular mystery species has stomata, right, on the sporophyte. Let's assume that you didn't observe it earlier, right? You did not put it under a microscope, right? And we didn't mention anything related to the microscopic observations earlier, right? But when you, let's assume that you put it under the mystery plant under a microscope, you observe actual stomata on the sporophyte. Now, presence of stomata on the sporophyte is not a characteristic feature of hepatocytes. It's a characteristic feature of either liverworts, not liverworts, hornworts, or of moss, right? So you can actually change. Definitely not hepatophyta. Now it should be either bryophyta or anthocerophyta, right? So like that, you actually can change because of the feature. The feature is actually presence of stomata on the sporophyte, and because of that, you change its phylum, right? You assign it to a different phylum. That's actually what we mean, right? By new character, where you change your phylum assignment based on that. Yeah, so you can come up with any other example, right? Based on the characters I listed. It could be presence of separate male and female plants, right? Which actually is again unique to just mosses. Right? Or if you observe that the sporangia splits across the middle, well, that's a characteristic feature only unique to hornworts, right? And anthocerophyta. So you can actually then again you can assign from your previous decision to a different decision. So those are actually 3 different examples that you can actually directly use, right? Now, if you want to actually be more ambitious, research yourself about the 3 phyla and pick other characters, that's completely fine. It's completely up to you what feature would make you to pick what feature that you are picking, and completely up to you to change the phylum assignment, right? So, and I provided you 3 examples already. Now, in terms of knowing, right, the characteristics I explained to the 3 phyla, that's good enough for you to know for the basis of knowledge for the purpose of practicals down the road. But if you research yourself, you will find a lot more other characteristics. But I don't expect you to know all of those details. This is Bio 122, not plant taxonomy. Any other questions? Okay, seems like we are all set. Emily, I hope my answer didn't confuse you any further. At least it helped you a little further. Okay. Now, if you don't have any questions regarding today's lab activity, exercise, or the homework, any question that you want to throw at me regarding the post assignment, right? So one thing I do want to mention, this is not particularly for one person or one group. I want to urge you to make sure, that the characteristics you list are actually specific to the family that is assigned to you, right? Don't, like, for example, if you are doing something on hummingbirds, right? Don't pick characteristic features that are unique to the entire order of hummingbirds, but just the family that was assigned to you. Don't list features that are common to all birds, right? So try to dig out information that are specific to the particular family you study, right? That's one thing I wanted to mention. Not that you did it wrong, but I figured that sometimes you list order level unique features as a family level unique feature, right? So keep that in your mind. So when you list characteristic features, start with family level unique features, right? If you run out of them, yes, then of course, then you can list order level features, but I can guarantee you, you can find enough features that are unique to the family. If you have trouble finding those features, you need to start talking to me. You need to tell me in that case, we have to have a conversation. I cannot give you examples, right? We can have a time set, like end of a lab, and have a conversation about, how to find additional features, where to find additional features. If you want clarification, hey, professor, is this particular feature unique to the family? We can have that conversation too, right? And another problem I have noticed is some of you struggled with determining what are the closest or related families to the family you picked. So here's how you do that. Go to Google for like look up what order your family belong to, right? And then do a Google search for order for that particular family's phylogenetic tree, right? Give me an example here. Jared, I'm asking you, I'm going to pinpoint you because you are my buddy. What order you guys studying? Can you remember off the top of your head? Anyone, like not just Jared, if he remembers. Jared,
I
what? Yeah.
don't remember the exact order. I remember the fish. Let me look this up.
Yeah. Tell me the,
Um,
yeah, what is the family? Just tell me the family name. That's fine for me.
Sturgeon fish.
Sturgeon fish. Okay. Okay. So let me show you what I'm doing over here in my screen, right? I'll start from the scratch over here. Some students actually ask this question, so I'm actually showing this to you, right? So I'm going to type sturgeon fish order, right? I want to know what order they belong to, right? And well, here we go. Acipenseriformes. Now what I want to do is actually I want to know the closest families of the sturgeon fish that was assigned to me. So I'm going to do, I'm go back over here. Acipenseriformes phylogeny, right? Or better yet, phylogenetic tree. Search for that. And then search for images, right? Over here, right? And then, so what over here, what you can look at is, you know, if you look at there, all of these are families, right? So, hey, Jared, I'm going to continue the conversation with you because, you know, we are looking at sturgeons. What exact family did we assign to you? The actual family name?
We had the tang sturgeon fish.
Blue Tang, I believe. Yeah, the family name. Let me.
Yeah, the
I can.
actual family name, like what day is it? I just don't remember. That's why I'm asking. I'm not trying to put you in, on the spot. What was the name of it? Anyone in your group, Jared, can remember that? In Jared's group?
I can type it in the chat. I'm not sure how.
Yeah. Like send it through the chat because you might not be able to
Okay.
pronounce it. Yep, got it. Right. Acanthuridae, right?
Oh, you posted it too. Sorry about.
Actually I just got it. And then what you have to do is, you know, look into these phylogenetic trees that we already have, and I'm struggling to see that particular family here immediately. It takes a little bit time to find Acanthuridae. Why is it so hard to find that particular family? That one actually didn't help very much. That does not do that either. Right. So for example, like what I did was, since I couldn't find anything directly by searching by the order, right? Then I, you know, sort of folded back, right, to put the family name, Acanthuridae phylogenetic tree. And actually that helped me a lot more than the previous case, right? Over here, this is the family that Gerard's team is looking for, right? And then immediately you can see two related families, right? Luveridae and Sansalidae, right? So So like that, you can immediately, in that case, figure out two closely related families. And if you keep looking over the examples, you might actually get better phylogenetic trees. So like that, either you can directly look up that particular order's phylogenetic tree, or look up the family's phylogenetic tree, right? Do both and you will actually get a good set of answers for that. And in certain cases, for example, when I was helping the hummingbird group, it was easier when I researched by the order name. But surprisingly, for the fish, I had to look up by the family name, right? So either way, that's actually the best way to look into that, right? To get information about the other related orders. I think I'm asking for 3 or 2 related families. So this is how you do that, right? And remember how you read a phylogenetic tree, right? Right. It is not the branch depth, it's actually the branch length, right? Which family has the closest branch length to the family you are studying, that is closely related, and then go by the next closely related one, right? So that's how you do that, right? So the mistakes I noticed is some students, some groups mention the closest related order or closest related genus, right? That's not what we are looking for. I want the closely related family. Usually family names, not usually, always. Family names end with day, right? The part day, right? D-A-E. That's actually the second thing I wanted to mention. The third thing I wanted to mention is the number of genera that you wanted to list. So this is not you actually, this is not somewhere you are going to always get a firm answer, right? Because we have splitters and clumpers, right? Some taxonomists, I'm a taxonomist myself, I like to describe different species. So I like to find subtle differences in species. So when we do research, we actually try to increase the species richness. And some people don't like it. They try to find characteristics to unite them. So because of these splitters and clumpers, right, certain people scientifically publish a larger number of species or a larger number of genera for a particular family, and others actually might publish a smaller number. And then again, the statistical applications we use to study phylogenetics, to study taxonomy, are quite different. There are so many different tools that you use. All of them are correct and all of them have certain limitations. So because of that reason, you will not get a single correct answer for the total number of genera in a given family. So don't kill yourself trying to find the one correct answer. You will not find one correct answer. If you find 3 papers saying like, this particular family has, paper A says 30 different species, genera. Another paper says 32 different genera. Another paper says 28. You can pick randomly any one of them. But if another paper says 50, another paper says 8, those 2 are clearly wrong, right? The one says 8 is probably outdated, right? The one that says 50 is probably erroneous or dubious, right? And another way to pick, like, how do I pick the correct one? Well, the most recent one is a better way to go, because most recent publication or the scientific study uses the most recent tools, technology for the research. So that's another way you can pick how to report the actual richness of that particular family. Those are 3 important parts that I do want to highlight. But again, everybody go back and take a look at the comments I provided to you. Go back and look at the overall guidelines to do the poster. Make sure you actually understand everything clearly. Remember, you are talking about a family. You are not talking exclusively about a particular species in the family. You are not particularly talking about the entire order. You are talking about a particular family, right? It's completely okay to provide additional information about a very cool example species or 2. We welcome additional information, right? And some students also asked me, can we show you a video? Absolutely, why not? Yeah, if you want to show another props beside your poster, like a video, pretty pictures, additional diagrams, right? Please do that. Just keep that additional prop in hand when you present it, right? And as long as that, yeah, please do that, right? Don't try to, if you feel like, hey, I cannot put all of this into the poster, right? This is going to suck. fine, don't put it into the poster. Have additional information as a prop by the side. As long as you can, commit, finish everything within like 10 to 15 minutes time. That's the time span. 10 to 15 minutes for the presentation, and we only have fewer groups. So if you go like few minutes extra, I'm not going to take points off, right? Don't take more than 5 minutes, beyond the limit, but you can successfully put everything into 15 minutes, right? So moving forward, make sure you follow the directions clearly. Ask any clarification from me. I prefer one-on-one meetings because I find always I provide a lot more feedback, useful feedback one-on-one. Virtually than through email. Always bring questions like, and discuss them after the lab so others can learn from you. And of course, practice your presentations and make sure who is presenting which part, right? And remember, you are presenting over the screen, right? So you can use index card to explain it, but do not read out of directly out of your poster, right? Because I can read what you can read, right? So keep a narration, keep your poster contents to the minimum we expect, and do reserve parts for your narration. Yeah, go ahead. Question.
So we'll be presenting a PowerPoint, like screen sharing it and talking over it?
You are correct. So you are only going to have a single PowerPoint slide, right? The entire slide will contain the entire poster. So yes, you will be sharing that particular PowerPoint slide as I am sharing my screen with you, exactly the same way. And then you will be talking about it. Yeah, narrating about it. Yeah, that's how it is going to happen. Yeah, and also what you can do is if you want to zoom right into a particular section, let's say, Mali, that you are presenting about the phylogenetic tree, rather than like having it on the slideshow, what you can do is, you can do something like this. Let me show that to you. Over here, right? So here I am actually right now not on the slide mode, right? Now I am on the slide mode, right? So I cannot actually zoom into certain parts of over here. Oh, actually I can, right? Let's see what I did. Go into the slide mode, right? If you click this little magnifying glass, right, over here, right, you click it, right, you can actually, navigate into certain parts, right? And then like zoom that in, right? Again, you can go to, if you want to like, show things over here, right? You can sort of zoom into certain parts. Another way to do that is actually keep it out of the slide view in this particular view. And you can actually increase, from the bottom, by this sliding bar, you can actually zoom into certain parts, right, of the slide you are showing, right? Like this. That's another way, right? And then, do this while you are sharing the screen. So that actually has greater flexibility to zoom into certain parts. So that's another option, right? Either you can actually go full screen and keep talking about the entire poster without zooming in, or you can zoom in the full screen, or you can go off the full screen and zoom in and out of certain parts. So there are so many ways to present it to the rest of the people. It's up to you how you do that. I don't care. I have no preference. Make it easier to your end. Good question. Anything else before we finish up? Alrighty, seems like I do not have any further questions. I will let you guys go. Bye-bye, guys. And I will stay around if any case, if someone wants to have a one-on-one conversation. But otherwise, it's good to go. You guys can actually say bye-bye.
Thank you. Have a good day.
Of course, Emily. Thank you.
Thank you very much. Have a great day.
Bye-bye, Jared.
Professor, I
Yeah,
have
James.
a question. So, did I ever get back to you about what the office said?
No, you didn't. Yeah, what happened?
So they said, they didn't really, that you didn't really need their signature.
Okay, good to
That you were
know.
fine to send it in.
Well, I will, no problem. That's actually, thank you for letting me know that back. Would you mind doing me another favor? Shoot me an email by the end of the next week.
All right.
Can you do that for me?
Sure. About
Yeah,
what?
just to juggle my memory. I have you marked. I was,
Alright.
yeah, I have you marked, so like I won't forget, but just, you know, I might. So just shoot me an email by the end of the next week, like a Thursday. Don't wait until Friday, Thursday, and I will make sure, because now I have like your mid-semester grade, right? I mean, and you're doing pretty good. So you're in my lecture too, right, James?
Yeah.
Yeah. So yeah, we'll do, buddy.
All right. So,
All right.
when will you have those grades in? Do you think?
Oh, all the grades up to now, I don't
Uh-huh.
I don't issue a midterm grade because you can look up, like if you go to Blackboard right now, you should see like all your grades in the Blackboard.
Right.
So it has your practical grade, it has your all homework assignment grades, first 2 exam grades, quiz grades, everything should be available to you.
Okay.
Yeah. What you don't have is actually, we cannot program a midterm grade like very effectively, because you know there is the weird way we do the final, and all of that complicates things a little bit, because it's the total out of many. So
Okay.
yeah, but other than that, everything graded, you know, is always available through Blackboard to you.
All right. So how would you send it in to UMass then?
Yeah, what I'm going to do is I will tell them your total possible grade up to now. Like, let's say you have taken 2 exams, 1 practical, that is going to be 270, right? Let's say that's all
Okay.
you have taken, right? 270.
All
And
right.
then let's say you got 90 in the first 2 exams, that is 180. And let's say 70 out of 70 in your practical, that is 280, 180 plus 70, 250. So that's going to be 250 out of 270, and I will convert that as a percentage grade and send it to UMass. That's what I'm going to do, simply.
All right. Cool. Thank you.
Yeah. What I don't have is, I don't have like a mechanism already displaying on Blackboard what your midterm grade is. I usually don't do that, and students usually don't ask.
Okay. Yeah.
Yeah, they prefer to wait until the moment, and there's a reason for that, because you know things
Yeah.
change. Yeah, but your case is not typical, so we can get around it.
All right.
All right.
Thank you.
Yeah, of course. All right.
All right. Bye. Have
Bye-bye.
a good one.
Yeah, you too, buddy.